Hi, everyone. Welcome to the Spark podcast, where we highlight innovation, science, and work being done to advance quality and reliability in laboratory testing. I am Nancy Duong, admin here at Solon Park Research Labs. Today, we're joined by Devon Vieira, a lab technician here. Devon works
Hello.
closely with our lab work and supports lab quality. And today, we're going to be talking about commutable quality measures, or CQM. Our software designed to help laboratories monitor quality control data more effectively, identify trends early, and she'll go more on in depth with that. So thanks for joining us, Devin.
Thank you for having me.
So to start off, I'd like to ask what industry problem originally inspired the creation of CQM? Like, why was there a need for something different in clinical chemistry quality monitoring?
Yeah. So it happened years ago when we realized that we needed to implement some sort of quality control data management system. We started checking out our options, which obviously led us to Biorad's program Unity. Um, but we realized so Unity is great. Unity has a vast amount of data, which enables quick and simple peer comparison, and that's invaluable. But it's only when you're using their controls. And we make our own high quality controls. So it didn't make sense for us to pay all that money for Unity when we wouldn't benefit from the peer reports. There's also the major costs of the software development, infrastructure, IT overhead. So it made more sense for us to spend that money developing a program that works for commutable controls. That part. That is why CQM is ideal for labs that run high volumes of third-party or in-house controls and have tight budgets or even niche analyte needs.
Okay, great. So for someone hearing about CQM for the first time, how would you explain that in simple terms, and why would it matter to the laboratories today?
CQM is a quality control data management system. One thing that it does is it stores all of your quality control values in one place, and it's a cloud program, so that data is accessible from anywhere. Another thing that CQM does is it graphs the values, which provides a visual representation of the data by plotting them. It plots the percentage bias of the tested control values from the manufactured assigned value over time. Um, so basically, it stores and transforms raw data into an easily interpretable pattern.
So you kind of touched on this earlier a little bit, um, but so one big difference is that, like, we can use like any kind of controls with CQM. So why is that commutability like so important, and it's such a great advantage compared to like the traditional like other QC systems then? Like maybe more touch on that.
Yeah, the other systems, other controls, they have a matrix effect change, and which makes them not commutable. Um, so commutability reacts to the full analytical system exactly as fresh serum or plasma does. Um, and it really shines when changing reagent lots. So normally you have to do lot to lot verification and validation. And when you're using commutable results, there's like basically no shift in control values. Um, it saves so much time. And there's also a harmonization among platforms. So our commutable material works for instruments manufactured by Beckman, Siemens, Abbott, Hariba, and more. Um, so it's just so accessible.
Nice. Um, so how does CQM help labs identify like where the issue is coming from? Like whether it's the reagents, calibrators, equipment, like how does it pin that down?
It's based on the pattern of the graph. So like how I said, it graphs the raw data, takes the control values, and it graphs them. So the pattern will signify either a trend or a shift. And a trend is a gradual movement upward or downward, typically over 5 to 7 points. And that shows a slow degradation of reagent or evaporation in liquid serum controls, even an aging lamp. So a shift is an abrupt jump to a different level. It can happen due to a new control lot or calibration drift. Um, or like reagent. It can happen with the when you change the reagents as well. And then there's also outliers or random errors, which are just scattered points that don't fall into a pattern. That can be like pipetting, variability between texts.
Can you share like a real practical example of how these trends would track in CQM and how it would help catch the problem before it affects the patient results?
Yeah. Um, so let's say there is a slow trend in your control values. Um, the, it may not be noticeable. If it's in control, it may not be noticeable to a tech, but the CQM can detect it and flag for a potential system bias.
Okay, so because the platform is cloud-based and an open platform, like how does that improve accessibility and flexibility across different instruments and lab environments?
It gives labs vendor neutrality. They aren't locked into one control manufacturer for all their instruments and analytes. Gives them freedom to use the controls that work best for them, including traditional quality control material or third-party commutable materials. It frees them from walking into very expensive contracts.
I think one thing maybe to bring up too, because it's cloud-based, like if you guys make updates and stuff, it moves through quickly then, right?
Yeah, it does. I mean, I've already made so many updates on CQM, and
Nice.
yeah. So like as for the program goes, we're always looking for feedback and how we can update it and improve it.
So looking ahead, like what future developments are most exciting for CQM? Like, do you see any like AI involved or more analytes, any deeper integrations or anything to look forward to?
Yeah, I mean, definitely. I think AI would be huge being integrated into CQM. I know that's something that we'll look into. For example, it can help with statistical processing, alerting, and reporting. That will lead to a better patient or a better user and patient experience. We're also planning on integrating surveys. This gives labs real-time feedback for proficiency in certain analytes.
Great. So, Devin, thank you for joining us and sharing your insight on CQM and the work being done here at Solomon Park. It's been really interesting and exciting to see how tools like this can help laboratories improve efficiency, and we get to be a part of that. And thank you to everyone listening, and we look forward to sharing more conversations from Solomon Park Research Labs.
Bye. Thank you so much for having me.